Replication and Transcription. So we're breaking up the different parts of subtopic 2.7 here. We're going to do translation as a separate lesson. This is a really big subtopic. So 2.7, we're going to focus only on DNA replication and transcription today, and we'll do translation in a later lesson. There's also HL extensions for all of these topics, which we'll be talking about in topic 7, and we'll disperse those throughout the SL lessons. So HLs, you'll learn a bit with the SL, and then we'll add more detail later on. And SLs, of course, this is all you have to know. No more requirements for you. So here are our RAS targets. I can analyze the process of transcription and DNA replication, and I can use specialized vocabulary to describe molecular processes. Let's start by looking at the structure of DNA. So remembering DNA structure from earlier, we have our bases that make up our gene, and the letters A, C, T, and G make up the sequence, the information, the instructions that tells the body how to make different proteins, what things it needs to make. So this is one half of a DNA, because of course DNA is double-stranded, and that's a single gene. Now the best way for you guys to take notes on this process is using this large foldable. So you have a really big foldable of DNA replication you should be using to take notes. It's posted on School. You get printed out for you in class. It's 3 pages attached together, and you can use it to help you. Now the ones with a star are the ones that everybody has to know, and the know in detail. The other ones are more like nice to know, extra kind of bonuses that are made good for HLs, or anyone if they were pursuing this field in the future. What I recommend you guys do is you color in the enzymes in the boxes a certain color, and then just put the description here, especially the ones with the star, DNA helication to write details of, and then match the same color on the full diagram, which we'll see in a little bit what that looks like. Okay, so DNA helicase is our first one. Here's DNA helicase. It's the subject of one of my favorite biology pickup lines, and it's going to unwind the double helix by breaking the hydrogen bonds between bases. Remember, our adenine and thymine have 2 hydrogen bonds to hold them together, and our cytosine and guanine have 3. So to unwind the helix, we have to break those bonds, and that's what DNA helicase does. Topoisomerase, this is another enzyme where every enzyme ends in ASE. These ones are going to relieve tension by unwinding the strand and cutting the sugar phosphate backbone. So the backbone is very rigid, and by cutting bits of it, it allows it to have flexibility to unwind the DNA double helix to then access it for replication. So this we have to write down in detail, but if you are HL or if you are interested, definitely write it down. But this DNA helicase is a must know and memorize for everybody. Okay, the next part of this application, step 2, is now we've unwound a little bit our helix. We're going to use the SSBPs, or single-stranded binding proteins here. These are going to prevent the DNA that's been unwound from rejoining again and naturally forming those hydrogen bonds it tends to want to form. I'm going to do that by just creating a block. So these are not a required one for SLs, but you should color them in and describe them. RNA primase, this is a big one for HLs, as well as polymerase 3. So primase looks like this, and this is going to make an RNA primer, hence the name RNA primase. And it's just a short sequence of 3 or 4 bases that helps get our polymerase enzyme started to replicate the DNA. Then the next one is DNA polymerase 3. Now, for SL students, you only need to know about DNA polymerase. You don't need to know the different versions of it. HLs, you need to know both. So this is the enzyme that's going to build off of the primers. So it needs RNA primase to lay the primers down, and then it will start adding the bases to match the bases of the existing DNA strand. So if there's an A, it'll put a T. If there's a C, it'll put a G. If there's a G, it'll put a C, etc. So again, pause the video if you need to and write this information down. You can paraphrase if you want, but these are important for HL in particular. Okay, the first 2 here are important for SL, and all 3 are important for HL. So we have nucleoside triphosphates. So these are our little groups of nucleotide bases. So these are the actual bases that contain our letters. Okay, they're the building blocks of the new strand, and because they're triphosphate, they contain energy for DNA polymerase to synthesize the DNA. So this is mine floating around in the cytoplasmic cell. These are my A, T, C's, and G's that are waiting to be added onto the DNA. The leading strand is the only strand that SL students have to focus on, and the leading strand is the strand that's going to be made in the same direction as what's called replication fork. So if you look here, the leading to this picture, it's going in one direction, and it's a continuous type of replication. SLs only need to know about the leading strand replication. HLs, you need to learn about the opposite of that, which is the lagging strand. The lagging strand is in the opposite direction, and therefore it goes in small little chunks. And we'll talk way more about why that happens that way when we talk about this later on. Okay, so again, first two important for SL and HL, last one's only important for HL students. Okay, so we've opened up our DNA, we've stabilized it, and now we start adding bases. Okay, replication step 4, we have DNA polymerase 1, and this is the next DNA polymerase that's going to come along, and those RNA primers that were set, it's going to replace those with DNA. So the RNA little bits that were there, it's going to replace them with DNA. Okay. We have DNA ligase. Again, this is important for HL, and this is the enzyme that's going to join all the DNA sections. This happens only on the lagging strand, so why only HLs have to know it. And it's going to glue. Ligate is to attach, so ligase is a gluing kind of enzyme. And the last one are our exonucleases here, and these are enzymes that double check there's no mistakes made in copying the DNA, so that way can leave, we're not producing any mutations by accident, any errors in the DNA copying. So all three of these are more for HL than SL. Okay, so here is the whole process. And what I want you guys to do, I'm going to go through and explain. This is the foldable you should have, and at this point you should have all of these boxes filled in, some in a lot of detail, some a little bit of detail, depending if you're SL or HL. So, first things first, we have DNA helicase, which is going to unzip the DNA double helix. Okay? And then we have topoisomerase, which is an extra enzyme that helps break cause small breaks in the phosphosugar backbone to allow it to have more flexibility to then unwind the DNA. Because before replicating it, we have to open it up, and this very tightly wound strands over here. So we loosen it up, and then we place our single-stranded binding proteins on each strand to prevent them when the DNA strands are close to each other from opening up. Now this opening here, this is a replication fork, and it's opened in this direction, so it's unwinding this way, okay? From 3' to 5' is the direction of the replication fork. Once we have everything open and stabilized, what we're going to do is we're going to start copying and trying to copy the DNA. So first we have RNA primase here, and you can see the brown colored bases. They're darker gray on your diagram if you haven't colored it in yet. Those are the RNA primers. They're going to what sets the bases to start. Now they'll be very similar to DNA, of course, except they have a don't have a deoxyribose sugar, they have a ribose sugar. And they have uracil instead of thymine. So they're different enough that cell can tell which one is which. And it'll start the section. Notice on the leading strand, it happens only once. On the lagging strand, it happens several times. So it starts, gets things going, and then DNA polymerase, which is, for SL students just call it DNA polymerase. You don't have to call it 3 versus 1, just DNA polymerase is fine. It's going to come along and bind to that RNA primer section, those few bases that are RNA, and then start adding DNA bases on, matching up the letters. So if this was an A here, that would be a T. If this was a C, that would be a G, etc., to make a complete copy of the DNA strand. At the same time, in the other direction, in the lagging strand, we're doing that same thing but in fragments. So RNA primase is making several little starting points, and then DNA polymerase comes along and copies for a little bit in that opposite direction. Then what's going to happen next is we're going to end up having DNA polymerase 1 that come along and remove those RNA bits, the primers, and replace them with DNA instead. So we have triphosphates that are both RNA-based and ones that are DNA-based, and we want to have the final version of our new copy of DNA strand as all DNA. To replace the RNA primers with DNA bits instead. And then we have DNA ligase going to come along, and it's going to attach or glue all the little bits together, the fragments together. Before the DNA is complete, and we can then go to the next phase in mitosis, for example, then we have exonucleases are going to double check and make sure no mistakes happen in the process. It's a lot of bits to remember here for this process. Okay, so there's a game you can play called, it's a Nobel Prize game, but you can actually try to make DNA copies and get you an idea of the practice application process. So the link is on Schoology. It's a good practice activity to do. You don't have to do it, but it's definitely a fun thing to do to practice and reinforce your skills. Okay, so let's review the enzymes for replication. So there's another diagram replication. There are many, many diagrams out there for this process. You can find one that makes sense to you and that you like. Definitely use the foldable because you can write all over it and it's nice and big. But you can also include some other diagrams, of course, in your notes. So in this diagram, we're looking in particular at the enzymes. So the enzymes that you'll notice are DNA helicase and DNA polymerase. These are the two that SL students have to know. So what does each of them do? Take a minute, pause the video, talk to your desk partner. Well, hopefully you have an answer at this point. DNA helicase, here it is in this one little triangle shape, is going to unwind the double helix, unzips the helix, so that way then we can have access to this each strand of the DNA and then start copying it and doing replication. DNA polymerase is what's going to actually attach those free-floating nucleotides as bases and match them up to make the new DNA. So in this strand, this is the old DNA, and this is the new that has one half of the old DNA and one half of the new. Okay. Now, what do you notice with the direction of replication here? Well, notice that it can only go, DNA polymerase can only work in one direction. It goes from 3' to 5', so it goes in this direction. And then because this strand is in the opposite direction and 5 to 3', what happens is you have DNA polymerase can only go a little bit in this direction and then it stops. It's like it's very fast at adding bases, matching them up, but it's not very aware of its direction, so it will go in the wrong direction for a little while, and it's like, wait a second, this is the wrong way, and it'll stop and start again, which is why the lagging strand does that in several small fragments. Again, we'll talk about that more with HLs later on. Okay, so polymerase only works in the 3' to 5' direction. And remember, we use 3' and 5' probably some DNA structure as the directions for DNA because left or right, up or down would make no sense. It moves around all directions. So 3' and 5' is referring to which carbon of the ribose, or in this case the deoxyribose sugar, that the phosphate group is bound to. Okay, so at this point we've gone through the process replication, and I want to spend a little bit of time talking about more kind of details of it and things the IB wants you guys to know. So, first things first, DNA replication, as we just learned it, is what we call semi-conservative. What does that term mean? Well, let's break it down. Semi means partially, in this case half, and conservative means you're holding on to, you're conserving, you're maintaining, you're keeping the same. Semi-conservative means that the daughter molecules from the DNA, when we copy it, we're not taking one original and making one completely new. We're taking our original parent, which is the blue strands, and with replication, we're going to get two copies now, but each one is half the parent and half of the daughter molecule. And this way, it's much more efficient. We're ensuring that we have matching the instructions on each strand, and we have less chance for big errors. So when we talked earlier about Watson and Crick and their famous 1953 article in Nature, we talked about the fact that they proposed structure of DNA, and when they stated, even their article, they stated that the complementary base pairing, A with T, C with G, that already implied a method of replication. They were already kind of implying that. There are several advantages to doing it this way. I already said some of them. Pause the video, talk to your partner. What are some of the advantages of replicating in a semi-conservative manner? Well, we definitely have advantages in terms of efficiency. We're not taking time to make two strands separately every time. We're making one, we're just kind of copying out the other and building the other half, which is a lot more efficient. And in terms of reduction of errors, so we're reducing the risk of mutation by having the original strand for each one. Okay, here's your first key ideas of this lesson. Take a minute, pause the video, write them down. If you have questions, now is the moment to ask a classmate, ask your teacher, get some clarification for yourself before you add more information. Okay, so let's talk about replication. So Meselson-Stahl is one of the experiments that the IB has chosen for you guys to memorize and understand. And this is where they confirmed that DNA's replication method was semi-conservative. And this was a really big deal at the time, trying to figure out a way to demonstrate that this was in fact semi-conservative. So what they did, like a lot of experiments in molecular biology, were trying to see things and analyze things that we cannot see even with a microscope at the atom and molecular level. What they did is they use isotopes that have different masses. In this case, because it's DNA, they use nitrogen for the nitrogenous bases, and they use nitrogen 14 and nitrogen 15. So remember that 14, 15 means the mass number of the isotope, which means that nitrogen 15 has one more neutron than nitrogen 14 does, so it's more massive, it's heavier. And if we look at the results here of this experiment, here's three kind of predictions. So if we have a semi-conservative model, then half the DNA is being conserved between one generation and the next, which means that after the first replication, if the parental is a heavier molecule, or different mass, the parental is pink, then at the first one, each DNA strand will be half of the parent and half the daughter. Half is conserved. One strand of each. And the next generation, then one will be the grandparent strand will still be present in two of them, and the next two will just be all new as well. In the conservative model, we have parental is left completely intact. For the first generation, half of it is just parental, half of it is just is all brand new. And the next generation, these ones will be both brand new, and this is one parent, and the parent one just continues on, and the data is the cells are just using that as information to copy the DNA. In the dispersive model, we have chunks of each one. So after the parental, a bit of it goes into each strand, and we have 4 strands that are a bit parent, a bit daughter, and the next generation we have even more that are a bit parent, a bit daughter. So in this case, none of them are all new, and none of them are all parent. So what did Meselson-Stahl do? Well, here's Meselson in the lab. What they did was they looked at the masses of the nitrogen molecules at different parts, different phases of replication of the DNA. Okay, so they're both still working in this kind of field. So Stahl's working at University of Oregon, and Meselson's working at Harvard. And they're both examining the peaks and the amount of nitrogen 14, nitrogen 15 in this DNA. So how did they do that? Well, we like to use bacteria for this kind of stuff. Why do we use bacteria so often in these kind of investigations in biology? The number one reason is bacteria are simple. If there's not a lot of organs or organelles get in the way, they replicate very quickly and efficiently, and it's easy to modify their DNA by just changing what's available to them in their environment to make their DNA. We know bacteria replicate by binary fission like most prokaryotes, and that when they do so, they're going to use whatever in their environment to make the new DNA. So they grew bacteria in the presence of nitrogen 15. So that way the new nitrogen, the new DNA they were going to make contained that heavier nitrogen. Then they transferred that bacteria to a solution containing lighter nitrogen, nitrogen 14, and they gave it enough time to grow for 1 or 2 generations. And then they look at the DNA in each group of cells. So each group of cells, they then spun them out in a centrifuge, so they're using the density of the material to separate them by spinning them very quickly. And the different weight molecules will separate into very distinct bands. So if we look at this diagram here, what we can see is at the beginning, we just had only heavy DNA, so it's a single band. All the DNA is separated, and it's one thing. That's the parent. Heavy DNA, nitrogen 15. One extra neutron makes a difference in mass. And then we transfer those cells to nitrogen 14 and give them enough time to grow for 1 generation. About 20 minutes is all it takes. Then we take a sample out, centrifuge it, and now we have 1 band. And this is hybrid. It's not the mass of 15, but it's lighter, but it's not the mass of 14 either. It's not quite that light. So this is showing that each one is half and half. Now this could be half and half because of this model, dispersive model, or because of semi-conservative model. But we know it's not conservative. That's been eliminated already. Okay, so if we go back to our diagram here of the process, the next thing we're going to be looking at is We do the next generation to confirm whether it's semi-conservative or dispersive. And by doing it 20 more minutes, allowing the next generation to replicate its DNA, now we spit it out and we have light DNA and hybrid. We have no heavy, so the parental's gone, but we have some that's just light and some that's hybrid. So this means because it's not all hybrid, we have the semi-conservative model of replication happening here. Okay, let's talk about PCR next, how we're going to copy DNA to do this. So PCR is called polymerase chain reaction, and this is how we make many copies of DNA. Now this is really important to be able to do a lot of genetic studies. We don't want to have a single molecule, a single sample of DNA, and then it gets used up in the lab and we have no more. We want to have a lot of copies to work with. So why would make a lot of copies of this? Well, what happens if you have a blood sample from an accident or a crime scene or something and you want to be able to analyze that DNA for evidence? Well, the more copies you have, the more likely you are to be able to analyze that correctly, and if something goes wrong or you do more tests, you haven't run out of copies. Same thing for someone doing a small sample of their DNA for a genetic counselor, for heredity evaluation, for 23andMe, they're curious, whatever it happens to be, paternity testing. Having more copies means we have more flexibility. So what PCR is it is a method that allows us to make many, many copies of DNA, and the tricky part here is that we're using not human enzymes to copy the DNA like we just talked about, but bacterial enzymes. And those bacterial enzymes that can work at a really, really high temperature that are called Taq polymerase. So Taq is a species of bacterium we're talking about, and they use this high temperature they can survive at. So take a look at this diagram and try to figure out what are the three main phases here: denaturing, annealing, and extension. Can you figure that out? Well, denaturing is we're going to increase the temperature to 95 degrees Celsius, and that's hot enough it breaks hydrogen bonds. The two DNA strands will then separate, and that does the work of DNA helicase, separates the strands. Annealing is we're going to lower the temperature down to 55 degrees Celsius and give time for these RNA, these primers, to attach. So annealing is to attach to, and we're going to allow the primers to anneal. The primers are put in there by the scientists. And then we're going to back up again to 72 degrees, and that's the right temperature for Taq polymerase, that enzyme, to then synthesize the DNA. And that's the extension phase. And it will make many, many, many copies very quickly through this process. So PCR is very useful for calculating DNA. Here's a little video clip of PCR to help kind of visualize this process for you.
A sample of chromosomal DNA, also called the genomic DNA, can be used as the starting material for the polymerase chain reaction, also known as PCR. With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest or target DNA is indicated as red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Taq polymerase. The primers are typically about 20 nucleotides long and are complementary in the sequence to the ends of the target DNA. Taq DNA polymerase is delivered from hot springs bacteria and can tolerate intense heat for the PCR reaction. A PCR reaction lasts several hours and typically consists of 20 to 30 repeating cycles. A cycle begins by heating the reaction mixture to 95 degrees Celsius. The heat denatures the DNA strands, breaking the hydrogen bonds that hold the strands together. After denaturing the DNA, the temperature is reduced to around 60 degrees so that the primers can form hydrogen bonds or anneal with their complementary sequence in the target DNA. Note that the primers and the target DNA follow base pairing rules. An adenine pairs with the thymine and a cytosine pairs with the guanine. In the next phase, the temperature is raised to 72 degrees Celsius. A Taq polymerase functions optimally at the temperature and begins to polymerize, adding nucleotides to the 3' ends of each primers attached to the DNA strand. After one complete cycle, there are two double-stranded copies of the target DNA formed. The PCR reaction mixture contains mainly copies of the primers and an abundant supply of nucleotides to perform many additional cycles. After a second cycle, there are 4 copies of the target DNA produced. After cycle 3 is finished, there are 8 copies of the double-stranded target DNA sequence, and note that only 2 of the double-stranded copies consist of just the target fragment, and the others also including flanking DNA regions. As the number of cycles increases, the products consist of greater proportion of the fragments with just the target DNA increases. After 4 cycles, half of the fragment consists of just target DNA and half of the fragments also contain flanking DNA. With each additional cycle, the number of copies of our target sequence doubles. At the end of 25 cycle, there are more than 33 million copies of this double-stranded target region found.
Okay, that animation was helpful for you to visualize the process. Here's what you need to know, key ideas. So check these, please. Any questions, please ask. Talk to a classmate. Email a question to me. Ask your teacher. Okay, let's do some practice questions. So here is a practice IP question, 8-point. We should definitely work on these long point questions for paper 2. So take yourself 10 minutes on a timer and do this question. Explain how DNA is replicated within the cells of living organisms. So pause, give yourself 10 minutes exactly, and go for it. Okay, we're going to look at a sample student response. You have your student response, and we'll look at a sample one as well to practice. So here is a sample answer. What do we think? How many points would you give that student? That student got 3 out of 8 points, which is, of course, not great. We want to do much better than that on these big point questions. So here's where their marks were achieved. They said the 2 strands are separated. So if you said that, that's great. The bases pair A to T and C to G, and the enzyme DNA polymerase builds the new strands. Things they lost points for is they didn't say the DNA bases are added. That was too vague. They didn't say that they said 2 new DNA molecules are produced in the process. That's also too vague. Talk about semi-conserved nature would be a good thing to say there. And 1 strand is conserved. You need to explain more how that actually happens. Okay, here are some tips for doing these kinds of questions. So when you're doing this kind of 8-point, bigger point question, some tips to try would include things like start longer questions with a simple definition of the process. So what is DNA replication in this case? Link structures and functions. In this case, lots of enzymes. So all of the enzymes to their function state. DNA helicase will unzip the gene to allow access to the DNA sequencing. That kind of thing. Be specific about the function of different things. Give precise details. Bases is too vague, but you could say things like adenine, thymine, guanine, cytosine. Give examples to help if you can with these bigger point questions, and avoid using general things like DNA is conserved. It's not the same thing as saying a dude strand shows semi-conserved replication. So be more specific if you can. Okay. All right. Here is another student answer. How many points did you give this student? The student got 8 out of 8 points. So notice how much more specific they are. They said DNA is a production of 2 DNA molecules from 1 original DNA molecule. That's a nice definition of the process, the replication is. In the replication, the first step is to unwind double helix, separate two strands. Helicase is anything that does this. That's perfect. They define the definition, the answer, the enzyme, and what it does, its purpose. They explained how adding nucleotides are added to each strand to make the double strands, how the base pairing happens, giving an example, explaining what enzyme does that. And notice again for SL, you just say DNA polymerase. Say 1 versus 3. Just polymerase is fine. How we have complementary base pairing, and there's one original strand and one new strand, each one after that. So we're going to keep practicing working on these types of IB questions throughout the rest of the course because your exam is coming, guys. Okay, the central dogma is this main idea of how everything gets copied in biology. So it includes replication, transcription, and translation. So we've gone through replication in quite a bit of detail here, making more copies of the DNA. We're going to start going through transcription now, and translation you'll do later on. But what is the purpose of these two things? Well, the purpose is to go from DNA to RNA to then build a protein. The purpose is to actually express the genes that are in the DNA to make a protein that the body needs for some reason. Okay, and how much DNA are we transcribing at one time? Well, we're not transcribing all the DNA. That would be insane. We're just transcribing one gene at a time, whatever gene is required for the protein the body needs at that time. It would be a waste of energy to do more than we actually need. Okay, let's talk through transcription. What's going to happen is, just like with replication and transcription, we're going to unwind the double helix so that way we can get access to the information on the code. And this is done by RNA polymerase, so that unzips in the case of transcription. So a different enzyme here. And we make an mRNA, or a messenger RNA, is made off what's called the template strand, which goes a 3 to 5 strand, using free mRNA nucleotides. So these nucleotides here are RNA nucleotides. They have a ribose sugar, and they have uracil instead of thymine. So remember, of course, the bases are different. They have the ribose and they have uracil, and that's making my DNA. Now, I'm not trying to copy the DNA. I'm just trying to read a message and just transcribe one part of the message to make one particular thing that is needed. So we saw this earlier of the gene in the template strand. How to change that to an mRNA? Well, the equivalent in mRNA is going to be complementary base paired, but I'm changing my T's to U's. So T thymine with adenine, adenine with uracil. Cytosine with guanine, cytosine with guanine, cytosine with guanine, thymine with adenine, cytosine with guanine, adenine with uracil, adenine with uracil. So that's what's happening in transcription. I'm going to show you another animation here showing this happening in real time. It's a really nice animation showing process transcription. Here we go. You see what's happening here? We're transcribing the gene and then barely under the gauge, just enough to get the information and then moving on. The yellow thing coming off, that is mRNA, the messenger RNA that has the gene that the body needs to make that our protein is trying to make. And RNA polymerase is that blue enzyme that will unzip it. Very, very fast. So for those who don't know, this actually using the yellow versus the green pieces, getting pulled in and being used to eventually, RNA polymerase will get to the end of the gene and release. It goes outside the nucleus for translation, we'll talk about later on. Okay, so take a minute, pause the video, and I want you to complete the strands here. So here is our DNA strand. Here's the top strand. What's the mRNA sequence for each of these ones here? So pause the video, try and figure out the mRNA in particular. We'll worry about the amino acids later on. So if this is my DNA, this is my mRNA. And here's the second one. Check yourself as well for that. Okay, here is another. Let's do some more practice with this concept here. So here is the question: outline the process which results in the formation of a strand of mRNA. Include details of how the genetic code is preserved. So 5 points. Give yourself 5 minutes to answer the question. Set a timer. Okay, so you've taken 5 minutes, you've written your answers. Take a minute and check your work. Here are the mark schemes. So these are the 8 points you could say to get those 5-point answers. So if you want to, you can ask someone else to check your work and exchange papers with a student. That can be a helpful way to practice as well. Okay, here is a sample student response. How many points did you give this student? Well, this student only got 3 out of 5, and this is why. They got points for saying the process is transcription. They got points saying RNA polymerase is the enzyme that's responsible, and they got points for saying description of mRNA is correct. But they didn't talk accurately about the process that makes RNA. It's called transcription. They didn't say mRNA. They didn't talk about uracil at all for the bases, and they didn't talk about the bases pair up in a special way that's too vague. Talk about complementary base pairing, talk about how it actually happens. For this kind of question, some tips. Start with simple definition, like we already said, link certain functions, and give precise details. Like bases pair up in a special way that is too vague. Talk about complementary base pairing. Okay, we are almost done with our big lesson here, guys. So the next thing is talking about the universal nature of the genetic code. So the genetic code is said to be universal because almost all life on Earth uses the same bases, same amino acids. This is very strong evidence for evolution. Why is that? Well, it means that every organism has the same DNA. Then some common ancestor had that first DNA and then was able to evolve and use that DNA, and then that DNA was used many generations. There's more than one way to communicate information biologically. This is just the one that evolved first and everyone kind of kept using. This universal DNA is really great for genetic engineering. So a great example of this is we can actually insert human DNA into bacteria. A great example when we do this is take the human gene for hormone, we put it inside bacterial plasmids, their DNA, and then that makes them produce human insulin. Why is this beneficial? Does anyone know what diabetics had to use before we could do this process? Well, they had to use pig insulin or other kinds of animal insulin. Most often it was pig insulin, which is both unethical for the pig, but also not the exact same insulin. Similar, but not exactly the same. So it's much better for the body to have the exact same one. All right, here's your last key ideas for today. And there is a big foldable, like when we have a replication for transcription and translation. It's a more detailed than the IB needs. It'll be really good for HLs, but if you're visual and this helps you, then you're welcome to have one. We'll make sure they're available to you guys digitally and or on paper in class. Okay, the last thing you can do is another practice activity, replication and transcription. You're going to use paper cutouts to figure out the directionality of DNA and the process of figuring it out. Once you've got it done, then ask your teacher to double check that you've correctly explained it. Okay? Use it for transcription as well. The more practice you do, the better off you're going to be. Guys, that is the end of our very long lesson, and this is even all of 2.7. So we're going to do the HL version of this process next, and then we'll come back to SLs to do translation later.